Hello everyone, Mr. Walker here. This is the third of three lessons on evolution. The first one was on the history of evolutionary thought, the second one on the mechanism of evolution by means of natural selection, and this one here, as the title says, evidence for evolution. And I'll start off just by acknowledging that there is no possible way that I could truly do justice to the amount of evidence that is available to support evolution. Huge amounts of evidence and from various diverse fields supporting evolutionary theory. What I'm giving you here on this slide is just a number of books, relatively recent books, and authors writing these books. Many books have been written on the evidence for evolution. So this one here, Richard Dawkins, The Greatest Show on Earth. Stephen Jay Gould unfortunately passed away a number of years ago. He wrote many, many books. This is one of them, Dinosaur in the Haystack. Neil Shubin talked about in his book here, Your Inner Fish, the transition from marine to terrestrial animals. Jerry Coyne, Why Evolution is True. And this last one here, American evolutionary biologist Sean Carroll, Endless Forms Most Beautiful, was one of his books on evolution. So the first one that we'll take a look at is the fossil record. So fossils, very, very dramatic exposure of fossils, like in this picture here. This is showing an Alberta fossil. In fact, it is called Albertosaurus. So very impressive when they are exposed, these organisms, traces of organisms, direct evidence of organisms that did exist in the past and obviously don't exist today. So this one here is going back a couple of 100 million years. The one on the right-hand side going back much, much further to about 530 million years ago. This is an organism called Hallucigenia. And where it is found is in what is referred to as the Burgess Shale, fairly close to Calgary, just across the Alberta-BC border in Yoho National Park, way up high on the side of the mountain. This is from what's called the Walcott Quarry in the Burgess Shale. And this is one of the very, very unusual organisms, one of the first animal organisms that would have existed again about 530 million years ago. So really impressive that these are found. So they are found in sedimentary rock. And you do need to, first of all, realize that the process of fossilization, it is incredibly rare. So that organism needs to die. It needs to be buried very quickly in an anoxic environment, which means free of oxygen. Can't be exposed to scavengers. And then it needs to remain in that area where geologic changes have not caused it to be lost over tens and possibly hundreds of millions of years. And then there needs to be erosion or some sort of mechanism that's going to expose that fossil. So again, it is going to be in sedimentary rocks, and the way that it does work with sedimentary rocks is typically in the oceans or ocean estuaries. Sediments that are suspended in the water are eventually going to settle down. And as they settle down, they form these sedimentary layers. So if there is an organism that has died, and if it is buried quickly, then these sedimentary layers could accumulate on it. And what that means is that the layers that are deeper down are going to be older, and the layers that are a little bit closer to the surface are going to be more recent or newer fossils. So the picture down at the bottom, what we see is that, well, eventually these do need to be exposed. So very, very rare that they're formed in the first place, and very rare as well, of course, that they're going to be exposed and we're actually going to discover them before the weathering process actually breaks down and that fossil is going to disappear from the record. So if we do have exposure, in this case here, it's due to a river or a stream that's eroding away the sedimentary rock. It can be due to other geologic events like uplift that's taking place, uplift from the oceans resulting in mountains forming. And that would be the case with the Burgess Shale, where we do have these marine fossils like Hallucigenia that ends up at the top of a mountain. And there can be other things, even human activities, landscaping, and digging can expose some of these sedimentary layers and the fossils. So in this picture here, we see that this one that is closer to the top, it again would be a younger fossil, and the one that's closer to the bottom or further down in the strata, that would be an older fossil. So taking a look at the sedimentary layers, ones that are deeper down are older, ones that are closer to the surface are younger, we can come up with a relative dating scale, and that just means one relative to the other, which ones are younger compared to which ones are going to be older. But how do we figure out how old they actually are? So how could I say that that hallucigenia is an organism that was around about 530 million years ago? Well, what they do use is radioactive or radiometric dating. And in order to do this, what they do need to find is within the sedimentary rock, they do need to find some radioactive elements. And where those are typically found is in volcanic ash. So when a volcano erupts and that ash is spread throughout the area, it eventually settles down. It contains radioactive elements, and it then becomes part of the sedimentary rock. So we see a layer here of volcanic ash, a layer here, and so on. So if we do find some fossils in between two layers of volcanic ash, if we take a look at these trilobites here, so if we can figure out the age of that volcanic ash up above, here 530 million years, and the one down below 545 million years, then we know that the age of those fossils in between has to be somewhere within that range. So now we have the fossils, we have the relative age, we have the absolute or numerical age of the fossils. When Charles Darwin was traveling through South America, he did come across fossils as well. It wasn't just living organisms, it was fossils. And he was kind of surprised that the fossils that he did find, they looked surprisingly similar to present day animals, or the animals that he found that were still roaming around. So he found a fossil of this one here, the Glyptodon, which is extinct. But what he found is that there are some living animals, the armadillo, that look surprisingly similar, and which of course are not extinct. So then he started to ask questions like, why is it that we would have these organisms that are no longer around today? Clearly, they were present in the past. The fossil record showed him that. And they're not around today, but yet the newer ones look surprisingly similar, but yet they're a little bit different as well. In other words, they have some modifications. So those are the questions that he began to ask. But of course, he did need to realize, of course, that there were some organisms around in the past that are not around today, and there are ones that are around today that were not around in the past as well. Quite often we hear about missing links or transitional fossils. So this slide here is showing the work of, well, one of those book covers that I showed you on the first slide. This is Neil Shubin at the lower right-hand side. And very, very impressed actually what he did. So taking a look at geology around the world, what he was looking for this missing link between marine animals, this one right here, and a terrestrial animal, this one here. So it wasn't really understood how this did happen. There was no transitional fossil showing an intermediate between the marine and the terrestrial version of it. And what Neil Shubin did is taking a look at geology around the world, he pinpointed a region based upon the age of the rocks where he thought it would be best to go and look for this missing link. And it turns out that it happens to be in Canada, northern Canada, in the Arctic, on an island called Ellesmere Island. So he did go there for several years, and eventually, well, he did find what he later called tiktaalik, which is the missing link. So if we do take a look at the picture down below, this is showing a pectoral fin from a marine animal. This is showing the hind limb for a terrestrial animal. And the question is, how do you get from one to the other? What is the evolutionary process? And what he did find is, well, again, this missing link here. So the marine animal, from about 385 million years ago, the terrestrial animal 365 million years ago, And radioactive dating places this transitional tiktaalik at about 375 million years ago. And this is just an artistic rendering of what maybe they think tiktaalik did look like. There are other really nice examples of these transition or intermediate fossils or missing links. So what this one here shows is the transition from terrestrial mammals to marine mammals. So marine mammals, whales like this one here, the baleen whale, the understanding is that it did evolve from not other marine animals, but from terrestrial animals. So on the left-hand side, we do see the bone structure of a terrestrial animal, and there have been some intermediate fossils that have been found. So if we just focus on a couple of things here, if we focus on the hind limbs and the pelvis, what we can see is that from the terrestrial form, which is usually described as something maybe along the lines of a like animal or possibly a pig-like animal, would be maybe similar to this one here, where we do have these hind legs. And then we see gradually a transition where we're seeing that they're smaller and smaller in terms of the hind limbs and the pelvis. And eventually we get to the point where they're really fairly rudimentary. They're disappearing essentially as we go from these terrestrial to the marine animals, to the marine mammals, like the baleen whale. So we do still have the pelvis and the hind limb bones that are there, but they're very, very small. They're not visible on the surface in any way whatsoever. So then the question is, well, why are they there? And the explanation is, well, that they did come from terrestrial animals, terrestrial mammals that did have these hind limbs. So keep in mind, this is taking place over a very, very long period of time, tens of millions of years, before we can actually have this transition that does take place. So there are missing links that are found. And all the time, they're kind of filling in these gaps in terms of missing links and transitional fossils. So another piece of evidence for evolution is to take a look at anatomy and comparing the anatomical structures of different organisms. So here we're going to take a look at mammals once again for this first slide. So the first kind of comparative anatomy is taking a look at what are called homologous structures. If we do take a look at the picture down below, what we have are four different mammals. So we have the human arm, the leg of a cat, the fin of a whale, and the wing of a bat. And if we do take a look at the bone structure, what we do see is that, well, yes, they are a different shape, but what I'm putting in the boxes here are all the same bone, and that is the humerus. Down below, your lower arm bones, the ulna and the radius, they're the same. Again, a different shape, but they're in the same location between the humerus and the next series of bones, which are the carpals, the metacarpals, and the phalanges. So what we're seeing in all of these cases is it is actually the same structure in each one of these four different mammals. But even though they are the same structure, quite clearly, they do have different functions. So in the case of the human arm, for carrying, the cat for walking, the whale for swimming, the bat for flying. So when we talk about homologous structures, they are the same anatomical structure, slightly modified, but they have different functions. So this is really important because what it does indicate is that they share a common recent ancestor. That is the case with these homologous structures. This one is exactly the opposite, analogous structures, and this is an indication of convergent evolution taking place. So organisms that do not have a recent common ancestor are exposed to a common selective pressure, and because of this pressure, because of natural selection, they have evolved similar functions, but yet they're coming from different structures. So the picture on the left-hand side, what we have is a flying squirrel from North America, sugar glider from Australia. Both of them would have a common selective pressure, and what that would be is avoiding the ground because that's where the predators are. So they have both evolved a strategy enabling them to go from one tree to another through the process of gliding. So a similar environment, a common selective pressure, did result in them coming up with a similar strategy, in other words, a similar function. But when you do actually take a close look at the structures, they're not the same. So different structures, similar function, and even though they might look the same, this indicates that they do not have a recent common ancestor. Picture on the right-hand side, we have wings from a mammal, wings from a bird, and wings from an insect. These as well are analogous structures. They do not have a recent common ancestor. Another piece of evidence that they like to look at is comparative embryology. There are some things that you can see in the embryo that are not present in the adult. So this is an interesting slide here. What it does show, and we could do this with a number of other animals as well, but it's taking a look at a chicken embryo and comparing it to a human embryo. So some things you just don't see in the adult human and the adult chicken, but you can see it, in fact, when you take a look at the embryo. So one of the things that it shows are these pharyngeal pouches, which give rise to gills. So we find those on the chicken embryo. Why are they there? We find them on the human embryo and other mammals as well. So why are they there? Well, what they do give an indication of is that they have a recent common ancestor. Well, in this case, probably not all that terribly recent, but they do have a common ancestor, and that ancestor would have come from fish, and that's why both of them do have these pharyngeal pouches. This one here, kind of interesting as well, a tail. Birds actually don't have tails. They have tail feathers, but they don't have tails, but yet there is one in the embryo. Some mammals have tails, but we don't, but yet there is a tail here in the embryo. So again, this is an indication that both birds and mammals and humans did have a common ancestor, and that would be the explanation for seeing these in the embryo. Next one I have is biogeography. So when Charles Darwin did travel around through South America, up along the western coast of South America to Ecuador, and then cut across about 1,000 kilometers or so to the Galapagos Islands that we hear a lot about in terms of the work of Charles Darwin. He took a look at the species that he found there, and what he found was, first of all, they're actually different on each one of the Galapagos Islands. The tortoises are a little bit different, the birds are a little bit different, but what they were closest to was the closest mainland, which was going to be Ecuador on South America. So the Earths and continents, they do kind of shift around due to plate tectonics. And this picture that's over at the right-hand side here, what it is showing is going back, and what we think was the arrangement of the continents about 250 million years ago, forming this large, massive continent called Pangaea. And what it does show is that right here, we have South America, and it was right adjacent to Africa. So that means that animals that we found along the eastern side of South America would be able to go back and forth between that region and what we now call Western Africa. So far enough back in the fossil record, they would actually be the same fossils. But of course, since then, the continents have shifted apart. So if we were to take a look at the strata at the more recent fossils, what we would find is that they're going to be different after the continents did separate. So this picture here, again, it's showing going back enough, far enough back in time. Taking a look at the fossil record, there would be the same fossils that were found in these regions of South America and Africa after the plate tectonics and after the separation of the continents. Then gradually these species and populations would become different. I mentioned this example here with Charles Darwin and the Galapagos Islands. So again, he took a look at what are called finches, and he took a look at tortoises, and he noticed that the finches were different on each one of the islands. He was told by locals that the tortoises, they could distinguish which tortoise was which just based upon what they looked like and which island they did come from. And if we take a look at a kind of more recent example here, this is showing a number of different birds called Hawaiian honey creepers. And on the different Hawaiian islands, there are different honey creepers. So it's really no different than what Charles Darwin observed with what are now called Darwin finches. But there would have been one original honey creeper that would have inhabited the Hawaiian Islands, but each Hawaiian island and each region on the Hawaiian Islands are a little bit different with different selective pressures. So because there are different selective pressures, what is going to happen with the original founding populations is they're going to spread out between the islands, and then based upon those selective pressures, we're going to have what is called adaptive radiation. Those populations on each one of the islands and regions within each one of the islands, they're going to begin to evolve independently of each other. And now what we have are a whole bunch of different birds, which are still honey creepers, but they look a little bit different. They now have adaptations that are going to be unique to that particular island. So going back to this one here, what Charles Darwin would then have thought was that those finches, where they originally came from, the founding populations would have come from Ecuador. Something would have transported them over to one of the islands, and then from there, as they went to the other islands, they would have encountered different environments, different selective pressures, leading to these different versions that we do have on each one of the Galapagos Islands. The next one that I have here is genetics or biochemistry or molecular biology. So in this case here, what they're taking a look at is directly the genetic information, the DNA, or they can also take a look at the product of the genetic code, which are the proteins that are produced. So proteins are made up of amino acids. DNA is made up of a genetic code of 4 different bases. They are abbreviated A, T, G, and C. So with genetic information, we can either take a look at the DNA directly or take a look at the amino acid sequences that are responsible for building the proteins. So picture on the left-hand side is just a picture of DNA, and we can see the letters kind of in here, the A's, the T's, the G's, and the C's. So what this can be used for is to determine the relationship between 2 or more different species and place them into what is called a phylogenetic tree of life. So the phylogenetic tree shows the relationship, the ancestral relationship between different organisms by taking a look at genetic information, either the protein amino acid sequence or the DNA base sequence. The picture at the left-hand side is taking a look at 8 different species, and it's showing just a very, very small portion of the DNA sequence for these 8 different species. So the idea behind this is we're comparing them. We're seeing how similar they are. And if they are really similar, then what we would say is that they have a more common recent ancestor. So if we take a look at A and B, for example, and take a look at every single one of those letters, what we can see is that, well, they're actually exactly the same. So that means that A and B, they would have a recent common ancestor, and that's what it's showing right here. That is that recent common ancestor. If we take a look at the 2 at the bottom, G and H, we can see that these, again, are exactly the same. So G and H, they would have a recent common ancestor as well. So those ones are a little bit different than these ones here. Okay, not much of a difference, but there's a little bit of a difference if you take a look at the letters, and I can see this one right here. Those are different. These ones are different. So what that means is that E and F, they have a common ancestor, but comparing E and F to G and H, their ancestor common ancestor goes a little bit further back in time. So as we go to the left in this case, this is going back in time to where they had a common ancestor. So that's using the DNA directly. The picture at the right-hand side, this is taking a look at one single out of tens of thousands of different proteins that make up your body and many other organisms as well. So this one here is cytochrome C. What they're doing is taking a look at directly the amino acid sequence and comparing a number of different organisms to humans. So this is saying how different is the chimpanzee in terms of its amino acid sequence for cytochrome C compared to humans? Well, it's actually the same. It's identical. There is no difference in the amino acid sequence for cytochrome C between a human and a chimp. So that's why we say that that is our most common living ancestor that we have is, or sorry, that we would have the most common recent ancestor with is with the chimpanzee. Ones that we're not quite as similar to, rhesus monkey, and then as we go further and further down, less and less similar to these other species that we have here. So the less similar it is, the further back in time, there would be a common ancestor. I thought I would just throw out a few misconceptions about evolution, and there are certainly more than what I do have here. So the first one, we of course do talk about survival of the fittest, but not necessarily the case. Do you really have to be the fittest of the fittest to survive? And I kind of like what Bill Nye, the science guy, had to say. He's not an evolutionary biologist, but he did write a book on evolution. And what he said is, you know what, you don't have to be the fittest. You really just have to be good enough to survive, good enough to reproduce, and then you get to pass your traits on. So it's not the best of the best, it's, well, just kind of good enough. Big misconception with number two, exposure to certain conditions leads to adaptive changes. Not adaptive changes that are passed on to the next generation. So this would be acquired traits. That would be Lamarckian that's passed on. So it's actually not the exposure to the conditions. You already have within a population variation, and there's already variation within the population that might allow some individuals to survive a specific selective pressure. But it's not the exposure that's causing the mutations, it's the exposure that's only selecting for individuals that already have those variations due to mutation, due to genetic mutations. Another misconception, evolution creates perfection. Absolutely not. All you need to do is take a look at a couple of examples like the human eye, and it is absolutely not a perfect design. And in fact, if you understand the physiology behind the human and mammalian eye, then you would appreciate the fact that it's actually probably designed exactly backwards compared to what would make sense. This one here, laryngeal nerve in the giraffe takes a very, very bizarre route in the giraffe. And the only explanation for that is if you do take a look at the evolutionary history of the giraffe, and that sort of explains the rather bizarre alignment and arrangement and the pathway that the laryngeal nerve does take in the giraffe. Humans evolved from chimpanzees. Well, no, they didn't evolve from chimpanzees. We would have had a common ancestor with chimps, but absolutely did not evolve from chimps. Acquired traits are not passed on. Misconception, acquired traits are not passed on. So this is very recent, but this is a new field of research, epigenetics, and what it talks about is that due to exposure to certain conditions, doesn't change the genetic code, but it might change how the genetic code is read. So things like exposure to stress, exposure to famine, doesn't change the genetic code, but it can change how it's read, and this can actually be passed on to the next generation. So in terms of what Jean-Baptiste Lamarck said, acquired traits cannot be passed on. Well, in this case, it is possible that some of those acquired traits can be passed on. Just one more thing here, are humans still evolving? Well, a couple of examples here. So natural selection does rely upon variation within the population. Remember that the variation is due to mutations. Mutations are continually taking place. It just has to do with whether there is a selective pressure that allows certain mutations to result in a variation which is favorable. And if that is the case, then the frequency of these varieties will change over time, and that's really all that evolution is. So just three pictures that I'll show you here. The one at the upper left-hand side, this is for a version of a gene in hemoglobin, which is found in red blood cells and is responsible for transporting oxygen. This version here is a mutated version, but it just so happens that having one mutated version, everyone has two copies of the gene, but having one mutated version actually protects people against malaria. So what we see is that in certain countries around the world, so Western Africa, Central India, where there's a high incidence of malaria, there's actually a higher frequency of this particular version of the gene, the sickle cell allele, within those populations. So those populations are a little bit different than populations in other parts of the world. So that is kind of a very small example of that, yes, human populations can change over time. And again, when we have a change in frequency of these alleles, that is evolution taking place. Lactase is the enzyme that is responsible for digesting the sugar in milk, which is lactose. And when humans did start to start animal husbandry, so raising sheep and goats and cattle, and they started to consume the milk, and the milk contains the lactose. Individuals that have this enzyme, they can break down the lactose, so they would have a selective advantage. So if we again take a look at a map around the world here, we can see that there are some countries where populations have a high frequency of this enzyme. And the explanation for that is these also happen to be the regions around the world where these people historically, over thousands of years, have consumed milk products, and those individuals would have a selective advantage, so more individuals within the population would have that particular version of the allele. The last one that I have here is the frequency of this one here. This one here provides protection against certain viral infections, and it just so happens that one of those viral infections happens to be HIV, human immunodeficiency virus, which is responsible for causing AIDS. So it's probably not the exposure to this one here that did eventually lead to a change in the frequency of this allele in these populations, but probably exposure to another virus which is similar. But what we do see is that in some populations in Northern Africa, there is a higher frequency of this particular allele. And what that also means is it provides some protection against HIV. So these people, even if they are infected with HIV, might be less likely to actually go on and develop AIDS. And the last one that I have here, so sometimes you hear about what is called intelligent design. Organisms on the planet placed here, not through the process of evolution by means of natural selection, but by some sort of divine being or divine force, and that's who we would call an intelligent designer. So evolution kind of says, no, there is not an intelligent designer, and it's just a process that does naturally take place over time and doesn't have any particular purpose or particular direction. But is there another intelligent designer? Well, if we take a look at some of these examples here, maybe we could ask the question, are we the intelligent designers, and are we changing evolution in an intelligent way? Well, the two examples that are on the left-hand side here, these fish, they're called glowfish, and these are pigs that we're taking a look at here, and their snouts and their hooves, they're actually glowing in the dark. And the reason for that is in both of these cases, humans have taken a gene from the jellyfish, and we've actually introduced it into the genetic information of these fish, these aquarium fish, and the pigs as well. So in this case, it's just kind of a novelty just to see if it can be done. But what it does show is that through a process of gene editing, which is improving all the time, we can actually make these changes ourselves. So again, you might want to ask the question, how intelligent is this really? Are there other things that we could do that are maybe a little bit more intelligent in terms of our modifying the genetic information of an organism in some beneficial way? Well, the picture on the right-hand side, this is the pig that's called EnviroPig, and what they did is they genetically modified it so when in its waste, it doesn't produce as much phosphates. So less phosphate in the urine means less environmental contamination, and phosphates in the urine going into the environment would lead to an algal bloom. So what they genetically modified was the pig, and they genetically modified the pig so it would not have as much phosphate in its urine. As it turns out, there's an easier way to get around this, and that's just to, well, feed the pigs different food, and that in itself can decrease the amount of phosphate. So again, possibility that we can genetically modify organisms and make it intelligent in some way, but again, I guess we need to sort of question whether either of these truly end up being an intelligent design.